Which one's the mommy chicken?
I know what to do.
Scientists still haven't found the exact cause of autism.
Wow! Oh, open that box.
But they do know that genes are involved.
Put the ball in the hole and give me the key.
One clue is the increased incidence observed in siblings.
Thank you.
If you have one child that's born with autism, what's the chance to each successive child that's born? And what we find there is it's about 15%, maybe even a little higher if it's a boy, maybe close to 20%.
An even greater incidence is found if the sibling is an identical twin.
What we see is that among identical twins, where one child has autism, 90% of the time, the co-twin will either have autism or be somewhere on the spectrum. That really suggests that it is a highly heritable disorder. The other part of that is 90% isn't 100%. So there's something else there. So autism is not purely a genetic disorder, but genetics does play a large part.
Humans have about 25,000 genes, and they exist in nearly every cell of the body. Genes are made up of linear sequences of 4 nucleotides, represented by the letters A, C, G, and T, arranged in long strands of DNA. These sequences act as instructions that are read like blueprint plans to make different proteins with different functions. Some define visible characteristics like eye and hair color. Some help with the biological processes like metabolizing the food we eat. And some help build and regulate the neural networks in our brain. One such gene, called the FMR1 gene, is located on the X chromosome. It makes a protein that helps build connections between neurons. Fragile X syndrome, a common form of inherited intellectual disability, can be traced to a mutation in this one gene. It is called a single gene disorder. But there is no single gene that causes autism. Over the last decade, researchers have discovered more and more genes associated with autism. Now hundreds of genes have been identified. Many of these highly implicated genes have one thing in common. They affect the development of the brain. When mutations occur in any of these genes, the regular production of their proteins and corresponding functions can become faulty. The length of a mutation can vary greatly. Sometimes just a single incorrect nucleotide in the DNA sequence can cause problems. Other times, longer segments of the genome are deleted, duplicated, or rearranged. These are called copy number variations and range in length to several million base pairs. Some genetic mutations are inherited. If a mutation exists in the parent's DNA, it can be passed to the child. But there is another type of mutation that has recently caught the attention of many researchers, a de novo mutation. This kind of mutation is one that does not exist in the regular DNA sequence of the parent. Rather, it appears anew in the DNA sequence of the child. It might occur in the generation of a sperm or egg cell, or it might occur around the time of fertilization. We have known for a while now that parental age is a causal factor. Older fathers are more likely to have a child with autism. The reason is, the cells that create sperm continue to divide over their lifetimes, leading to increased chances of a mutation appearing.
As cells divide, DNA mutates. And one thing that we've known is, for example, sperm, as men get older, if you look at 70-year-old men and 60-year-old men and 50-year-old men versus 20-year-old men, there are more mutations accumulating in the older sperm. It's kind of like, you could think of it this way, that as you get older, things don't just heal as well. If you hurt your knee, it takes longer to heal, etc. Your skin cut, etc. Same thing with DNA repair. DNA is always being replicated and, in dividing cells, and mistakes occur. And sometimes it's not repaired well, and that's essentially what's happening.
Parental age is just one of many environmental factors researchers are looking at. These factors not only include the air and water around us, but a wider range of elements.
Now, we're still learning what environmental factors might contribute to autism, and the research is casting a very broad net. We do believe that these factors will have affected the very early development of the child, so we're interested in the prenatal environment. This could be everything from what the mother ate during pregnancy, whether the mother had an infection, and also whether the child was born prematurely, what happened during the birth process, and then right after birth, also during that early postnatal environment. Was the child exposed to toxins? So many potential factors that could be contributing to risk for autism.
What is the relationship between the environmental factors and the genetics? What is the interplay between them? How is it that these factors can change or modify the way that genes are expressed?
Another very important area of research that's emerged over the last several years is an area called epigenetics. What we know is that the environment can influence how genes are expressed.
One of the main epigenetic mechanisms is the coiling and uncoiling of different regions of the DNA strand. When the DNA is uncoiled in an open state, it is exposed to transcription machinery, which can then find its correct binding site and initiate expression of a gene. This is turning the gene on. The gene can be silenced by a number of chemical changes that make the DNA strand coil up in many successive layers into a highly compact form known as chromatin. The genes in this area cannot be expressed as their binding sites are physically hidden from the transcription machinery. The genes are effectively turned off.
What's happened over the past 20 or 30 years has been both a genetic and a neurobiological, neuroscience revolution in terms of our understanding. The first 25 years of autism research, I think, was characterization of the disorder, mostly on the psychology side, and then a lot of studies saying what it isn't. And to a parent, saying what it isn't is not very helpful. It's not immunization. Okay, what is it? And then the sentence stops. So I would say that the more we say what it is, the more we embrace this idea that it's a complex interaction between genes and environment, we're getting closer to understanding what those interactions might be, and to working more rapidly towards things that we can do right now in terms of interventions, the more the relationship and the strength will build between families and between clinicians and between scientists. And I think that's where advances are going to take place at a more rapid pace than they're taking place now.